This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 860, recorded on January 28th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despomier.
Hello, Vincent, and everybody else. It's a very interesting. Do I have my mic still? My mic is on, yes.
It is.
I can barely see the second tower of the George Washington Bridge from my window today because it's snowing. And we haven't had snow in a long time, so we're due to get a lot more over the next couple of days. So you know that the temperature is below freezing, and it's kind of a grotty, gray, snowy day. That's it.
Wow. You're supposed to go down to minus 12 on Sunday. That's Celsius.
Minus 12.
It's been cold. It's been cold. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Right now, it feels like minus 12 Celsius. It's actually minus 7 Celsius, which is 21, feels like 10. And it's sunny. There's some puffy clouds. So it's pretty nice.
And from Austin, Texas, Rich Condit.
Hi, everybody. 59 and sunny. I'm sorry, 57 and sunny. Let's be precise here. Headed for 59. Approaching freezing overnight. 34 degrees is the forecast. We're going to have another freeze next week, according to this. Fairly normal.
I don't know if we'll get to it, but we have a letter from someone in Austin explaining the weather.
That's right. And the citrus fruit.
It must be a pretty long letter if you're going to explain the weather.
It is pretty long, and it has no virology in it. It's all about the weather. It's very funny. But all right, here we are. Kathy, do you have a PSA for us?
Oh, do I even have the Google Doc on the right monitor? No. Okay, now I do. Yes, there's a vaccine town hall scheduled for Monday, January 31st, 2022 at 7 p.m. Eastern Time. If you want to sign up for it, go to asv.org/education. Or if you want to encourage somebody who has questions about vaccines, asv.org/education.
I have a PSA from Columbia University Medical Center where there is a job posting for an assistant manager of the BSL-3 laboratory. And I will put a link to this job posting. It's for the Aaron Diamond AIDS Research Center. So it's called a High Containment Laboratory, HCL, assistant manager, to help manage the center's BSL-3 facility and to help with SARS-CoV-2 experiments. So you have to know how to do some experiments and also have some organizational skills. There's a whole list of responsibilities. And if you are interested, there is a way to apply at the link. I think I'm going to apply. See what happens. Just kidding. Now, we advertised a job for Columbia some time ago so this is part of environmental health sciences. It's their purview, right? These BSL-3 labs. And we advertised for a person to be working there, and we got someone, and she came, and it worked out really well. So maybe they can get someone. The Aaron Diamond AIDS Research Center, of course, is headed by David Ho. And it moved not too long ago from another location. All right, so there'll be a link in the show notes, as well as a link to asv.org/education. Today, I wanted to do the Wayback Machine because older papers, this is not that old actually, but it's only 2005, I think, right?
Yeah.
But it could be a world away. It's just, and it's a really important finding because it led to the ability to make mRNA vaccines. And it was published in Immunity. And I have to say, this is back in the day when it was just a paper. There's only 4 authors, first of all, and that's not a big deal in itself, but it's, what is it, all black and white, right? Yeah. No color figures. There's no supplemental data, as Kathy has noted. And it's got the old look of just, it's the title and 2 columns of text. There's no graphical abstract. There's no huge lists of oligonucleotides or anything else, all the other stuff that's been added. It's just 4 authors and science. Boy, I really like this.
I have to say also that it's extraordinarily well written.
It is.
I really like this paper. It's so clear and so straightforward. They've just done a marvelous job.
Suppression of RNA recognition by toll-like receptors, the impact of nucleoside modification and the evolutionary origin of RNA. So important findings, as you'll see, that led to the ability to make mRNA vaccines. The authors are Katalin Carrico, Michael Buckstein, Hu-Ping Ni, and Drew Weissman from the University of Pennsylvania School of Medicine. It's published in Immunity. And we don't have any co-first, co-second, co-third. There's just corresponding author, Carrico.
And these 2, the first and corresponding authors, have been, prominent in the news, and rightfully so, in the development of these messenger RNA vaccines. This is just a first step, an early first step in this whole thing. But I'm so glad you picked this, Vincent, because this is just a marvelous paper.
Yes.
I don't remember where it came up, but it came up somewhere in a recent discussion, and I clicked on it and I said, oh boy, we have to do this, because I always wanted to do these. I have to say, it didn't hit my radar. It did not hit my radar. You can't look at everything, but I noticed that it's just hard to look at everything. So this is all about how nucleic acids are sensed. In the introduction, they do a very nice job summarizing, in our cells, in eukaryotic cells, there are sensors of foreign, or PAMPs, pathogen-associated molecular patterns, which can be protein or nucleic acid. And the toll-like receptors, as you know, recognize nucleic acids. And they're both DNA and RNA sensors, and they talk about them specifically. And the idea that pathogens have different nucleic acids, different enough so that they're sensed as foreign, because we have nucleic acids ourselves, right? For example, in some DNAs, there is methylated C and CpG. So when there's CG, 2 bases, CG next to each other, the P is the phosphate in between. The C can be methylated, and that's how the innate immune system can recognize that as foreign. And they say in the beginning, we don't know if RNA is subject to the same kind of modification, right? Would modification of RNA affect its ability to be recognized by an innate sensor? And of course, once a molecule is recognized, then the cell makes cytokines and it kicks into the inflammatory phase to try and react, right? So for example, I didn't know ribosomal RNA is 80% of cellular RNA. I didn't know that number. Maybe I forgot it. Ribosomal RNA has way more modifications from mammalian cells versus bacteria. Ribosomal RNA has 10 times more pseudouridine and 25 times more 2' O-methylated nucleosides than bacterial RNA. Whereas this is interesting, the ribosomal RNA from mitochondria has very few modifications, and of course, they were once bacteria, right? Or mitochondria.
Right. Even going before that, they kind of talk about the historical misunderstanding. For years, bacterial and mammalian DNA were portrayed as having the same chemical structure, which hampered the understanding of why only bacterial but not mammalian DNA is immunogenetic. So even going back to DNA, it's like you would have said DNA is DNA is DNA, no matter where it's from. Right. And then, as Vincent's just saying now, we now appreciate that ribosomal RNA, is different between humans and bacteria and so forth. It's a real key point to this whole thing.
In particular, tRNAs are heavily modified. and mammalian tRNAs, up to 25% of the bases are modified. And there's much less modification in prokaryotic tRNAs. In fact, they say bacterial mRNA has no nucleoside modifications, whereas mammalian mRNAs have modified nucleosides like 5-methylcytidine and others. And interestingly, modified nucleosides are more frequent in viral than in cellular RNA. So they're establishing this idea that maybe modifications make a difference for recognition, right? You don't want to recognize yourself, so maybe you modify your nucleic acid or not. So in the case of bacteria, the bacterial DNA is modified, but not ours. And so in this paper, the approach is to take various RNAs, and many of them are made by in vitro transcription using a T7 RNA polymerase promoter, which you can hear Rich talk about in a very old video when I visited him. He was talking about the experiments that his buddy, Ed Niles, was doing at the time while he was cooking dinner for us. It's funny. It's a funny video. So they make RNA in vitro, but they also extract nucleic acids from different sources, and they put them on cells, and they look for a readout of sensing as a cytokine production. We'll talk about that. Yeah.
And the gist of it is, although they don't ever quite come out and say it directly, that when they're making these RNAs in vitro, if they're making them with the standard bases, there will be no modifications. And so that's their sort of ground zero RNA. But they can artificially include modified bases, and then that will make something different.
So in the first experiment, they isolate RNA from different compartments, cytoplasm, nucleus, and mitochondria of eukaryotic cells. And this consists of total RNA, tRNA, mRNA, and then they mix it with lipofectin, which is a reagent that facilitates the nucleic acid getting into the cell. And then they put it on monocyte-derived dendritic cells in culture. So dendritic cells are an important sensor of various molecules. So you don't want to work with Vero cells, right? You want to work with relevant cells. So this is a very good thing to do. And later they'll actually look at dendritic cells from blood. So monocyte-derived dendritic cells, and then they add these different RNAs with lipofectin, and they're measuring the secretion of tumor necrosis factor alpha, TNF-alpha, one of the cytokines that would be produced when a cell senses that there's some nucleic acid there. And then, so they say all these mammalian RNAs induce TNF-alpha secretion, but very low levels, especially compared with RNA synthesized by T7 transcription. The figure is really very nice.
Yeah, striking.
TNF production on the y-axis, and then on the x-axis, the different RNAs, and they do plus and minus RNase, an enzyme that would chew up the RNA to show that the signal is RNA dependent, right? And so, for example, poly-IC, a synthetic polymer of inosine and cytidine, which you can buy, you put it in cells, it's sensed very nicely, and you get a production of tumor necrosis factor. And the negative control is just lipofectin by itself, right? Very important. You need a negative control, make sure that's not doing anything, and it doesn't induce any TNF-alpha. And the positive control is a toll-like receptor 7 ligand. It's an agonist, something that will tickle in the TLR and make it lead to the production of cytokines. And indeed, it's called R848, makes a lot of TNF-alpha, and of course it's resistant to RNAs because it's a protein.
And that just tells you the receptors are working.
Yeah, it's your positive control. Then they have an in vitro transcribed RNA, which they have made. It doesn't really matter. Unmodified, right? Gangbusters of TNF induction, which all goes away when you digest the RNA with nucleases. And then a series of different mammalian RNAs, total, nuclear, cytoplasmic, poly-A. They induce a little, but not very much. The only one that goes up quite a bit is mitochondrial RNA. tRNA is very low as well. And then E. coli total E. coli RNA induces quite a bit, so it's sensed. But tRNA from E. coli is not very much anyway. Right, so that's quite interesting. And right, just by looking at it, you don't know why, but the suggestion might be, well, the transcript is unmodified, so maybe that's part of it, right? The nucleus they use is called benzonase. Anybody ever heard of benzonase?
Yeah, I used to use it.
Used to use it?
Yeah. I think the magic of benzonase is that it's at least modestly detergent resistant.
I see.
So you could use it under circumstances when there was detergent around, and I guess it's both a DNase and an RNase. Is that correct?
Yeah, single-stranded RNA and double-stranded RNA. Well, I don't know about DNA, but both single and double-stranded RNA.
Somehow it was used before my time in adenovirus preparations. I remember hearing about benzonase, but never had to use it myself.
Anyway, the conclusion is that they make, which I love, they're making a conclusion in each experiment, which really helps. Here on TWiV, it helps us because we can understand what they think it means if we don't. These results show that there seems to be an inverse correlation with the extent of their nucleoside modification and how much they lead to the production of tumor necrosis factor.
So to say that again, the more modified the RNA is, the less immunostimulatory it is.
Right. And the in vitro transcript, which is not modified at all, is the most stimulatory next to their positive control, right? Okay, so nucleoside modifications might influence the ability of RNA to activate dendritic cells, so they decided to do this systematically now. So they're doing in vitro transcription reactions. So you have a DNA template with a T7 RNA polymerase promoter, and you make RNA in vitro. And this is so funny. You're making like nanograms of RNA, but for the mRNA vaccines, they made kilograms. I just love pointing that out all the time, you know. So there's two ways that they do this. They do transcription in vitro with nucleoside modified NTPs, right? With which you can get, and you can mix different modified bases into the transcription reaction. And then they have human 293 cell lines that produce individual toll-like receptors, TLR3, 7, and 8. And in this case, they're looking for activation by nucleic acids by IL-8 release, a different cytokine. So if you put these unmodified transcripts into these TLR3 cells, you make lots of IL-8. And interestingly, if they modify the transcript with M6 methyl on the 6th position of adenine, or S2U, no detectable IL-8. Those two modifications really completely knock it down. But other modifications, 5-methyl-C, 5-methyl-U, pseudo-U, and 5-methyl-C pseudo in the RNA had a less remarkable suppression or no effect at all on activation.
So just a moment on the chemistry of these, because I'm always interested in the chemistry. So two of these, 5-methylcytosine and 6-methyladenine, are simply the normal nucleotides that are methylated on the base,
right,
at one position or another. I had to look up S2U, which is a thiolated uridine, thiolated in the 2 position. So that means that on the number 2 position on the uridine ring, which is otherwise unsubstituted, there's a double bonded sulfur. Okay. And then the pseudouridine is the most interesting to me because we've talked about this before. That's simply uridine, where some enzyme called pseudouridylase, after the thing is made and incorporated, comes along and plucks that base off of the sugar and rotates it 180 degrees out of around a sort of a one axis and plunks it down again using a carbon 5 instead of the one nitrogen. And the interesting thing is that this preserves the base pairing capacity because there's, the two double bonded oxygens and an amino and a ring nitrogen are in the same place. But it otherwise modifies its appearance and recognition by other enzymes. I remember, so the first science project I ever did in a laboratory independently was in Harry Noller's lab at Santa Cruz. So this is over 50 years ago, was to try and figure out the purpose of methylated nucleotides in ribosomal RNA. And unless we get into an Easter egg or something here, I won't bore you with the details. But I was taking biochemistry from Harry at the same time. And I remember a quiz that had pseudouridine on it, some sort of a question. I wish I could recover the question because I flunked it. But Harry was on the ball with that stuff.
Did you figure out the function of the methylated bases in ribosomal RNA?
I figured out that the hypothesis was wrong. The hypothesis was that the methylation would serve as recognition sites for ribosomal proteins to bind in the course of ribosome assembly. So, okay, you asked for it quickly. We made tritium methyl labeled ribosomes by feeding E. coli tritium methyl labeled S-adenosylmethionine, which is the methyl donor for the methylation of ribosomal RNAs. And then took those and lightly digested them with, fixed them in formaldehyde and lightly digested them with ribonuclease and ran them over a Cephalex column where the protein-bound RNAs would come out in the void volume and the unprotein-bound RNAs would remain included, and asked whether the distribution of methyl groups was different in the two fractions. The hypothesis being that if ribosomal proteins recognize the methyl groups, that most of the methyl label would come out in the void with the proteins.
Didn't.
Not true.
Didn't, yeah.
50 years ago, I remember it in exquisite detail.
Yeah, of course. It's
including doing the formaldehyde fixation in a tris buffer and having everything go bonkers. And it turned out to be really acidic. And it turns out that tris buffering is with primary amino groups. And formaldehyde reacts with those. And so it takes the tris no longer as a buffer.
Interesting.
Okay. So I had to change to a phosphate buffer. I learned a lot.
I'm sure. But you wanted to learn. Right?
It's true.
A lot of people.
And mostly I wanted to screw around in the laboratory.
Dixon, do parasites have modified bases in their RNA?
They must. Yeah, malaria has lots of them, I think. And they've got AT-rich genomes and all kinds of other crazy things that prevent you from easily dissecting them. So, yeah, they do.
So in this section, they use the cell lines making individual TLRs, and they find that basically nucleoside modifications limit the activation of TLRs, the various TLRs in these cell lines. In particular, M6A and S2U are really good at suppressing the production of cytokines, whereas of and they work on TLR3, which is interesting, whereas other modifications work through TLR7 and 8, which is suggesting that the modifications, right, are interacting with the TLR in some way. Then they take these same cell lines, again, making different TLRs, and they isolate RNA from natural sources and put them in, look for IL-8 secretion. And the summary here is that RNA that is scarce in modified nucleosides from bacteria or mitochondria, right? So bacteria and mitochondria are scarce in modified nucleosides. They stimulate particular TLRs, human TLRs. But mammalian RNA that has a lot of modification are either minimally or non-stimulatory. So bacterial RNA is not us, so it comes in and it would be recognized and stimulate cytokine production. And our own, with the exception of mitochondrial RNA, our own RNA doesn't stimulate very much because it's modified. Does RNA come out of the mitochondria very much? Does anyone know?
I was thinking about this because they have a wrap at the end of this paper about whether necrosis or apoptosis stimulates an immune response. And they talk about the role of mitochondria in that.
Right.
And I forget the details. Maybe we can go into this. But the point is that if cells mitochondria are coming apart, the cells know it.
Right.
Okay. Because of the RNA. And I've thought about this. What is it? There's a role for cytochrome C.
It stimulates. Right? If that comes out of the mitochondria, the cells go into apoptosis. Right.
Yeah. So my sense is that cells have developed ways of knowing that there's trouble in the mitochondria.
Yeah, I agree.
Okay. Yeah. By responding either to cytochrome C or mitochondrial RNA.
Yeah.
If your mitochondria are in trouble, it's time to take yourself out of the game. Okay?
That's a good way to put it. I like that. That's really good. It's true, because if you can't make energy, you're in trouble, right? And I think it's an ancient response,
Gotta be.
Yeah.
Gotta be. So, and I think, obviously, as you say, mitochondria are evolutionarily derived from bacteria. So you could say that this is just a holdover from that, but I'm guessing it's more than that.
Yeah.
Okay. That over the course of evolution, the cells have used that difference, among other things, to make sure that their mitochondria are okay, or that they, shoot themselves if they aren't.
Because if it were deleterious, it would have been gotten rid of, over the years, but it's stayed. So it's an interesting point. Yeah. All right. So next they look at dendritic cells, which are going to be, they're antigen presenting cells. They're big sensors of stuff, and they have a lot of TLRs. So can these RNAs activate them, and does it matter if they're modified or not? So they have RNAs of different lengths and different sequences, which they add to dendritic cells. These are, initially they use the macrophage derived dendritic cells, monocyte macrophage derived dendritic cells. And they get different results with the different RNAs. They say 25% of the time the presence of M6A reduced activation more potently than M5C or pseudouridine. And this kind of variability they don't see in the primary dendritic cells, which they get from people, as you see in a minute. And they do a northern blot, which we were talking about not too long ago. I was saying many people don't do them anymore, but they used the northern analysis to show that the modifications don't change the stability of the RNAs once you put them in a cell, right? Because one trivial explanation would be that they're just getting degraded, and maybe that's why they're not sensed, but they're there. That's good. It's a good control. And I'm sure they had no problem doing that, because this is in the era where people were still doing northern blots a lot. And then they get dendritic cells from the blood, which they can differentiate or divide into two populations. There's monocytoid and plasmacytoid kinds of dendritic cells. There are many more types, but these are two that you can get from blood. They produce TNF alpha when they're exposed to the TLR7 agonist. Only one of them. So the DC1 is the monocytoid. Only that responds to poly-IC, which they say at a low level, which they say shows there's not any toll-like receptor 3. And 2, which doesn't respond, because TLR3 recognizes poly-IC. And again, putting transcripts in both cell types, that induces TNF alpha secretion. And if you modify the RNA, transcripts where the U is replaced with M5U, pseudo-U, or S2U are not stimulatory, where the RNAs with M5C and M6A were almost as potent inducers of cytokines as unmodified RNA, which is interesting, right?
Yeah, and the last two lines on this are interesting, whereas they take an RNA where the RNA has both M6A and pseudouridine.
Yeah.
That is not stimulatory, but if they take an M6A RNA that has only M6A in it as the modified nucleotide, and another one that has only pseudouridine in it,
yeah,
and use that it is stimulatory. Okay? So, I guess I don't know quite how to parse that out.
They think that means the DCs have something else that's sensing besides the TLRs that they know about, right?
Yeah.
Yeah, something about those two modifications being in cis.
Yeah.
Yeah.
Okay, so this what we see is that M5C, M6A, pseudos, S2U, others, they decrease. Oh, sorry. In addition to this data, so when a dendritic cell gets a PAMP, it becomes activated, makes cytokines, but it also puts on its surface various molecules, including MHC molecules, which are all part of it being activated because it's going to go on and maybe interact with a T cell in the lymph node. So these molecules all go up, and the modified nucleosides inhibit the upregulation of these surface molecules, which is a really nice way to look at something other than cytokine production. I thought that was good. And they do that by fluorescence-activated cell sorting. Okay, so how much modified base do you need to do this, to inhibit? Gosh, that could have practical applications, couldn't it? What's the minimal frequency of any single base that is needed? So what they do is they do in vitro transcription with different amounts of the modified base versus the unmodified, right? So you could increase the modified and decrease the unmodified base. And they show that, in fact, when you do that, you get the expected proportion of modified bases depending on what you put in. So if they, between 0.2 to 0.4% of M6A, pseudouridine or M5C in the RNA, which is about 3 to 6 modified nucleosides per one molecule of the RNA, which is 1,500 bases long, okay? 3 to 6 modified bases per one RNA is enough to cause inhibition of cytokine secretion. If you do 14 to 29 modifications per molecule, it gets about half the capacity to induce expression. So that is showing you a direct relationship between the number of modifications and the amount of inhibition of sensing of the molecule.
So importantly, it doesn't have to be 100%.
Right.
It can be just a relatively small fraction. And there is that proportionality. The more, the better.
They also chemically synthesize RNA oligonucleotides, and they show that these are shorter oligonucleotides. Now, at a single base is enough to abolish the activation of sensors. A single base in an oligonucleotide. It's really quite amazing, right? So all of this shows
those molecules are about 21 or 31 long, so they're really short.
Really short. All right, so stimulation by RNA is suppressed proportionally by the number of modified nucleosides. So that's the paper. Those are the data, and there's a very long discussion about, what it means and the evolutionary implications of it. But they say one thing that I think is really interesting: effective nucleoside modification on RNA immunity has not been explored. Nobody really has looked at this. And you've heard the story of Catalin, who wanted to do this and couldn't get support for it, and ended up collaborating with Drew Weissman, who also thought this was important, right? And so they both did this series of experiments, which is pretty cool. By the way, if you're wondering, there are hundreds of different types of modified nucleosides in RNA. Hundreds, not just, the handful that we've looked at here. And pseudouridine is the most abundant one in RNA. They go on to talk a bit about that, and I have a picture of pseudouridine in the show notes. You can see uridine, pseudouridine, and then there's a derivative called N-methyl pseudouridine, which it turns out is what is in your mRNA vaccines. This turns out to be a very potent inhibitor. And in fact, they replace U in transcription in vitro of the vaccine mRNA. They replace U with N1-methyl pseudouridine. And there have been a series of papers after this, a great summary review article in Frontiers in something. Frontiers in Cell and Developmental Biology, the critical contribution of pseudouridine to mRNA COVID-19 vaccines. I think pseudouridine needs to be a household word, don't you?
I think so. Everybody ought to have a portrait of pseudouridine on the wall over their couch.
So they talk about the history of all this, including this paper that we've talked about. But then, of course, Carrico went on to say, and they indicate this in the paper, actually. I think it's the very last sentence.
Very last phrase.
So why is this good? This could advance our understanding of autoimmune diseases. So some autoimmune diseases, you make antibodies to your nucleic acid. They said maybe this could help. Maybe it would help us understand why bases are modified in RNA. And then give future directions into the design of therapeutic RNAs. Yeah, there you go. And
it has not escaped our notice.
It has not escaped our notice, exactly.
They don't say that, but we can read into it that's what they're saying.
So they went on to publish a series of papers looking and focusing in on pseudouridine and what it was doing. Eventually, they put it in mice, right, and showed that the pseudouridine makes it far less immunogenic. And they start to get this idea, hey, maybe it could be used for mRNA vaccines. And then they also look at, it turns out it's more resistant to certain RNase degradation, and it's translated more efficiently. RNA with is N-methyl pseudouridine, which is all now making sense, right, for the vaccines, because apparently they make a lot of protein, and they are not recognized as foreign because they have this methyl pseudouridine in it. So, well, this really leads to a lot more work over the years, which then at the beginning of the pandemic, BioNTech and Moderna take advantage of, right? It's just, and I think Carrico ends up consulting for BioNTech, right? And
yes.
What did Moderna do? I forgot. How did they get into this? Did they take licenses out from someone? UPenn or something?
I think so, something like that.
I don't remember.
Definitely both companies got in on it.
Yeah, which is a wonderful story. And I bet this gets a Nobel Prize at some point because it's really it's basic science leading to an amazing clinical effect, right? Saving the world, the shot that saved the world. This book.
Right.
Mm-hmm.
That's got the whole story in it about the role of these individuals, and it'll answer some of those questions about who did what when.
Greg Zuckerman, we had him on.
And I just forget all the details.
There's a recent trial that I just learned about where they're now trying the same technology for HIV.
Yeah.
Sure.
That just arose on the radar screen.
And influenza virus too, sure. I mean, maybe it's a solution to the kind of poor VE of influenza vaccines.
Well, I can't imagine that the messenger RNA technology for vaccines it won't trigger a Nobel somewhere down the road.
Yeah, sure.
How they actually parse this out and who they give it to is another question, and that will be very interesting. But these two ought to be high on the list. And the list can only be 3 long, right?
These two have to be on it because they did the fundamental science, right?
Yeah.
And then maybe you have one person from, I don't know.
We had a discussion last week about how, the Nobel decisions can surprise you, put it that way.
Oh, yeah. But I think this is, as everyone may know, Carrico had a hard time working because people didn't, they didn't like this. And it's just,
people just have this idea that RNA is so fragile that how could you use it?
So what's the lesson we have to learn? Don't be so skeptical. Let people do things, right?
Right.
And see where they go with it. Allow them to do them.
It's easier to predict the past than it is to predict the future.
Science is particularly vulnerable because you have to get money to do it, right?
Tell me about it. And
your university doesn't pay you to do. They don't give you money to do experiments. You have to get it from NIH or elsewhere, and it's hard.
But we had a guest on recently that played the saxophone and didn't worry about it as granting because he didn't have to apply for any. So if you work for the NIH, you're in a better position.
It's better, but you need to be productive because they have to.
Of course,
it's the intramural money. They have to apply for it periodically, right?
I was going to make the point when John was talking about that their programs do get reviewed, and I actually participated in one of these recently, and it's a rigorous review.
Oh, yeah.
And if your work isn't up to snuff, you hear about it. Okay, and sometimes something gets done.
I knew someone who worked at Mill Hill, and that was the same there, only even more rigorous, because everybody got reviewed exactly every 5 years, and if you didn't produce over those 5 years, you were asked to leave. It was that simple.
Yeah, I thought the NIH intramural reviews were every 5 years.
I think that's right.
5 years.
They may be 5 years. And there's usually a lab head who then has laboratories beneath them, right, or with them, and they kind of write the application based on data that everyone gives them.
That's right.
Yeah, but the review is an in-person review of the lab head and the sub-lab heads and postdocs and graduate students. Find out if they're happy with their environment.
Yep.
And what kind of people they are. So it is, actually a review that includes how are you using the facilities, how are you using your money. Mm-hmm. Okay, so it's comprehensive.
I think you need to be lenient with science because, with an NIH grant, if you apply and they don't like it, you're done. They cut you off. They don't give you, we'll give you a couple of years to see if you can fix these issues. No, you have to apply again or you're done. It just seems draconian, right?
How about the Salk Institute?
Excuse me, Dixon. That's a fundamental difference in these reviews. These reviews of the internal program have to do much more with what have you done than what is it you're telling us you're going to do. Okay, and that's a trap with the extramural system, that we've discussed before. Really, if you're going to evaluate extramural research programs, maybe the emphasis ought to be on, have you made a contribution reliably over time, rather than, are the details of the experiments you're proposing going to work out? Because you don't know. That's what we're talking about.
There's some politics in the term productive. So that you can say, they've produced trivial research, like, can you use mRNA as a vaccine, for God's sakes? Who needs that? They wouldn't dare say something like that, but they might think it. Or they might, out of jealousy, just not support somebody that's actually doing great work. And I've seen examples of that as well. Nothing's perfect.
I wanted to just go back one more thing here. There's a very nice paragraph from this Frontiers paper that summarizes. So in a follow-up study published in 2008, Carrico et al. Proposed the inclusion of pseudo-U would be the crucial step for mRNA to mature as a therapeutic tool, both in gene replacement therapies and in mRNA vaccination. They confirmed that unmodified RNA compared to pseudo was more immunogenic in mice. However, Carrico et al. Also suggested that while pseudo-modified mRNA could be preferable for mRNA vaccines, it would eventually require the co-administration of an adjuvant like LPS or an immunostimulatory oligo. In this regard, it appears that lipid nanoparticles played this immunoadjuvant role as both carriers and adjuvants for the approved COVID-19 vaccines, mRNA vaccines. Just wonderful. I love when basic science just informs all of this, right? Mm-hmm. So there are many more papers, and so starting in, it's just even a little bit before 2005, there are some earlier papers that they cite that set the stage for this work. So very important stuff. All right, let's do some email. Let me take this first one from Lori. I wrote to you yesterday, but I just have to add today that I listened to your interview with Julie Youngner episode 373. Wow, love it. I laughed and cried. Who would have thought a science podcast would cause such emotion? What an interesting life. He died the year after this interview. So glad we have this record. Thank you once again. So I put this in the queue because, that was just wonderful that I was able to do that. I went to Pittsburgh and interviewed Julie, and he had an incredible career. It was fascinating, and he opened up and talked about it. And I want to relink to it because all the new listeners should watch it. All the old TWiVs are kind of buried in the shadows of evolution, the dust of evolution. But there's some really good ones out there. And then he died a year later, and his family said, "Can you give us we're so glad you did this because now we have a record of him." Right.
Mm-hmm.
Very cool. And I also had interviewed another older, not virologist. He was at UConn, and he had done some very early work on interferons. And then he also died not too long after his TWiV. Can't remember his name. And then when I asked someone else to be interviewed, they said, "I don't think this is a good idea."
There's a trend emerging.
Let me try and find his name. UConn. Yes, I bet you know who he is.
No, I had the wrong name.
Philip Marcus.
It was Philip Marcus. Okay, I Googled that and came up with somebody at Berkeley in mechanical engineering, so that was wrong. But yeah, Philip Marcus. As soon as you said Connecticut and interferon, I thought that's it.
So I drove up there and did the podcast in his office, and I had parked in the garage. So I went to get my car, and they only took cash in the parking machine. I don't carry any cash. So I went back in, and he gave me $10. I said, "Okay, I'll mail it to you." And I never did. Then he died, and I felt so bad that I never mailed him his $10. Sorry, Marcus. But it was a fun interview. So that's why I put this in, because there's a lot of old, previous episodes that are really good. And nobody's done any of these. Nobody else does this. TWiV does it, long form, etc. And then we just mention the next one because this is polio. And Anthony sends a link to an article, and his email subject was Post-Polio Now. So this is a link to an article, The Polio Experience. It's a speech for a Rotaract banquet, October 2011. So it's a speech. And he mentions, Neil Young had polio.
It's actually a she.
She.
Mavis Matheson, MD.
So the speaker is Mavis, and she's talking about Neil Young, right?
Yeah.
So Neil Young had polio in 1951 during the Ontario outbreak. And the song Helpless, which apparently they played at this, the song you just heard was inspired by Neil's childhood experience with polio, and Joni Mitchell sings backup. She was 8 when she was among the 1,200 cases of polio in Saskatchewan in 1952.
I'll be darned.
And like me, the speaker says, I also had polio in that outbreak.
Isn't that amazing?
It is.
And so the song,
who would have known?
The lyrics, she then speaks the lyrics and then explains the references to polio. There is a town in North Ontario. I can just hear the song, right?
Yeah.
In his falsetto. Neil contracted polio in 1951 in Ontario. All my changes were there. And then she puts in parentheses, all my changes, meaning polio. Blue windows, parens, lying in bed, paralyzed windows were our only connection to the outside world. Throwing shadows on our eyes, shadows, fear, pain, and the unknown. Helpless, 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 which is the chorus, right? How it feels to be paralyzed with polio. The chains are locked and tied across the door. Isolation ward. Wow. And so I thought that was pretty cool. I didn't know any of this.
Neither did I.
Wow.
And if you look at the link to her page, this is Mavis Matheson, MD. Had polio in 1952.
Oh, there she is.
Left leg was severely wasted. Neck and shoulder weakness. She, in quotes, recovered completely and led a full and busy life until she developed post-polio weakness and fatigue 40 years later. practiced medicine until 1993. Today uses a power chair outside her home, rests and spends time with her partner, sons, etc. So post-polio syndrome, Vincent, tell us about that.
Yeah, so Anthony's email thing is, and post-polio now. So the question is, will Neil Young develop post-polio? So the observation is, it's kind of relevant to long COVID, right? Because people who develop polio, often you could regain the function of limbs by therapy. Often, not always, but often, and have a normal life for many years. And then some years later, 20 or so years later, you would start to become paralyzed again. So, and often many people who had post-polio ended up in a wheelchair again. And what's going on? There's no virus present, but it seems to be the gradual loss of neurons with age. It compounds what you already lost with polio, so you're unable to have limb function. But it's not 100% of patients. I think it's less than 50% of polio patients develop post-polio. So Neil may not, who knows, right? But it's, I think, 20 to 30 years after your initial. So 51, wow. He's, well past that, so he may have dodged it. But I had no idea that song was about that. It's great, right?
Neil Young is 76 years old.
Wow. Interesting.
Yep, very interesting.
Kathy, can you take the next one?
Mary writes, hello, good people of TWiV. Sweden is not recommending that children 5 to 11 be vaccinated, according to the linked article below. They say the benefits do not outweigh the risks. Are the risks really greater when vaccinating this age group? I thought the data show the opposite. I'd like to hear the panel's take on this, as this concerns me because my twin 7-year-olds have already received their first dose and are due for the second one next week. Your podcast has been my number one go-to for all things related to SARS-CoV-2. Thank you for your service, Mary. And she sends a link that I haven't looked at, but I can just say, cumulatively from listening to Daniel on the clinical TWiV updates, that the benefits of the vaccine do outweigh the risks. Children are not at no risk for COVID-19, and the benefits from the vaccine certainly outweigh any of those risks.
My sense is from both Daniel and conversations that we've had recently as well, is that the manufacturers are, they're always cognizant of the side effects, but particularly so in this sort of circumstance. I mean, we talked last time about how apparently the 2 low-dose regimen that they have tried is not sufficiently immunogenic enough for them to be confident that it's working. So rather than go to a higher dose, they're talking about putting in a 3rd dose. And to me, a lot of that is about let's keep the risk low.
Yeah, I think Daniel, in the last update, someone asked the same question. They asked him if he would vaccinate his child less than 5. He said yes. Immediately he said yes.
Yeah.
So I don't know what Sweden is thinking, but I don't agree with that. If I were there and I had a kid, I'd get them vaccinated.
So I found one statistic that more than 2.6 million kids under the age of 12 will have had their first vaccine shot by the end of someday in mid-November.
Mm-hmm.
And if there were serious risks, we would have heard about it.
That's
even
right.
if they were at a tiny, fraction of that 2.6 million.
Yep.
So, and it's way more since then, I'm sure. So,
yeah, that's a very good point. Rich, could you take the next one?
Kevin writes, Dear TWiV crew, I've been enjoying the recent TWiVs, and as of late, you seem to be hammering the message that RNA load from RT-PCR does not correlate well with infectious viral titer. In other words, that RT-PCR CT value is not a good proxy for the level of infectiousness. While I now agree with those statements, I have to admit I was quite surprised when presented with the data that supported them. Specifically, in Figure 2 of the Puhatch et al. preprint, there is a statistically significant trend and yet a very poor correlation between RNA genome copies per mil and focus-forming units per mil. Many previous studies rely on the false premise that RNA load is proportional to infectious viral titer, but since that premise is incorrect, the conclusions they make may be in error. For example, you all have said this is one reason why the Provincetown study is flawed, the study that showed similar CT values between Delta-infected, vaccinated, and unvaccinated people and jumped to the conclusion that they transmit at equal rates. I'm curious then on your opinions regarding other studies that rely on this same false premise. Namely, one, the University of Colorado study that suggested 2% of people carry 90% of SARS-CoV-2 quantified by RNA viral load. Two, the Singapore study that suggested vaccinated people clear virus faster than unvaccinated people quantified by viral RNA load. Three, the contentious pre-pandemic EcoHealth Alliance study that many people would argue was gain of function by showing that WIV1 chimeras replicated faster, again, according to the false premise that increased RNA load is proportional to infectious viral RNA titers. That is not to mention the other flaw in the argument that this work demonstrates a gain of function, that there is not a t=0 time point for the RNA load kinetics. See this article for the most pertinent figures from EcoHealth Alliance. Report to the NIH and Vincent's quote that this is no question gain of function. Perhaps he has a different opinion now. Gives a link to that study. I could go on and list more studies that rely on this false premise for their conclusions. But I guess my greater interest is in hearing your opinions on when we should and should not dismiss conclusions that rely on it. Do we dismiss them before translating our conclusions to the public health policy? Do we give them the benefit of the doubt in the midst of the pandemic? Do we dismiss the conclusions as paradigms for virology, but err on the side of caution for public health? How much virology has relied on this sometimes false premise? Thank you for reading us. Thank you for continuing to explain all of pandemic's nuances to me. Best wishes, Kevin, who writes from Wisconsin, where it is snowy and minus 12 degrees Fahrenheit. Ooh, 12 degrees Fahrenheit is mighty cold. Minus 12, that is. Okay, so there's a lot in this.
I thought it was a really good letter.
Yeah, very good. And I can't off the top of my head necessarily address all the particulars, so we could get into them. The thing that strikes me more than anything else, and then I'll pass this on to others, is that there is a great tendency to try to think about all of these things in black and white. That things are absolutely true or absolutely false, okay? Where the truth is often somewhere in between, okay? And I think what's important is to know that these studies are out there that suggest that there may not be, oftentimes, a strong correlation between RNA copies and infectious virus. And to be able to incorporate those things into your judgment of a scientific study in total, okay? I mean, the virus does have to make RNA to reproduce, okay? So it's not like there's nothing going on, okay? But it's not necessarily a strong proportionality. So understand what the data are, and then try and use that to nuance your evaluation of studies. Beyond that, I can't do these individual things.
Yeah, I think that's a good point. There is a trend, right? In the study from Switzerland that we did, right? Remember, the R squared was pretty poor between infectivity and RNA, but there is a trend, right? But it's not a great correlation.
So, for example, the Singapore study suggested that vaccinated people clear the virus faster than unvaccinated people, quantitated by viral load, okay? In that particular case, you can't necessarily correlate the RNA virus to whether or not they're infectious or what their viral load actually is. But if you're comparing two different populations of people and you see a difference in the dynamics of how much RNA they have, that to me is worth considering. Okay, that might mean something.
Well,
two populations are different.
I think that would say, okay, this suggests X, but if you want to prove it,
yes,
you should look at infectivity, and that's what the Swiss study did, right? They actually, but I would agree that they did multiple time points as compared to the Provincetown study where they did one. And I think if you look at the Singapore data, I did it with a class yesterday. The first time point in vaccinated and unvaccinated patients with PCR is exactly the same level, right? Whereas if you look at infectivity, it's different. So,
I think a way to think about it is the RNA load data may be suggestive, but you need some orthogonal, independent, rigorous way
I like that.
to show that.
Yeah, in fact, remember the Boulder study? We did that on TWiV a long time ago, where 2% of people have 90% of the RNA. I think it's suggestive, but it doesn't prove it. They weren't able to prove it because they had the samples and they did PCR and so forth, and they couldn't do infectivity. But ideally, you would want to do this. So that's how I look at it. You make suggestions and then you have to prove it.
But I do have to admit that when people are giving a talk or something and they say, this is not statistically significant, but it's trending toward, but even more when they put it in a paper, it's like, then it's not statistically significant. You are just playing the odds wrong.
That's right.
Yeah, it's true. So this quote from the EcoHealth, so I said it's no question gain of function. So the Intercept got the grant application from EcoHealth, and they put it out there, and they sent it to me to look at. And you can find it at that link. And so there are two experiments there. There's one where they're basically taking a coronavirus from bats and putting different spikes in from other coronaviruses to see, if the spike allows them to infect cells differently. So there are two kinds of experiments. One is a growth curve, but they don't measure infectious virus. They do PCR. And I said, can't make any conclusions. And there's no time zero, which even makes it worse. And it's a bar graph on top of it with a couple of time points, right? So I agree, there's no time zero, and Kevin picked up on that. But the other experiment is weight loss in mice. And that's what my quote was based on, because some of the chimeric viruses clearly made mice lose weight faster. That's a gain of function. But then there are also some viruses, some of the recombinants don't cause mice to lose any weight. That's a loss of function, right, with respect to weight. So that's what my quote was based on. So I don't want to have a different opinion because I thought the PCR experiment was bogus, but that's what most people were basing their gain of function on that. And I don't think you could conclude anything. And the lady talking to me just couldn't get that because she thought that the RNA load by PCR was definitely infectious virus. She could not wrap her head around that. She asked me like 10 different times and I tried to explain it to her. But I think the article reflects that. So that's where that comes from. But Kevin, good job for picking that up. Very good. Dixon, this is perfect timing because this email is for you.
Yes, it is. Richard writes, for Dixon, the Associated Press tweeted, Walmart said it has taken a stake in agriculture startup Plenty, becoming the first large U.S. retailer to significantly invest in indoor vertical farming. Vertical farmers say their method brings higher yields while using less water and land. Well, a lot of other people say that too, by the way. You don't have to be a vertical farmer to say that. I'm very pleased to see that some large outlet stores have gained interest in this, and Target is another one that's also partnered with another vertical farm company called InFarm. There's a lot of activity out there, let's just put it this way, and it's hard to keep up with it. So I appreciate people writing in like this, but unfortunately this tweet has nothing to do with virology, and I wish it did.
The pictures of the technology here are fascinating.
Oh yeah, Plenty is a great company. You'd like just going through their website. It's quite wonderful. They're located in San Francisco.
Cool.
Dixon, you and I visited a vertical farm in Newark. Do you remember?
We did. We went to AeroFarms.
It was an indoor farm, right?
They're doing really well right now. Doing very well.
And also, we went to the one in Brooklyn. What was that? It was on top of a Whole Foods, right?
Oh no, that was
something Greens.
No, Gotham Greens. Gotham
Greens.
That's right. There's so many there. They're running around in my head like little fairies. There must be a couple of thousand vertical farm companies now in the world. So, looking back to 2010, when there weren't any, this is quite a remarkable accomplishment.
Man,
it must be that the climate really is changing.
I go to the Plenty website, and there, right in my face, is Plenty baby kale.
Yes.
Couldn't they pick something else?
Well, it's a high iron.
Am I going to have to eat kale for the rest of my life?
No, you won't. But you'd enjoy the baby kale, because what you think is kale right now, I'm pretty sure you haven't had something like that. I had a similar reaction, Rich, and when someone handed me some and I actually ate it, I said I would like some more. So, I'm not a big kale fan, but I think it is a very healthy.
Arugula, lettuce, kale.
That's right.
Kale. All
kinds of kale. But they're on, and that's the other trend that's also occurring now, because people really like this food. They want to see more of it, but they also want to see more variety. And so a lot of these big companies like AeroFarms is now experimenting with blueberries and raspberries and cherries, and not cherries, I didn't mean that. It was strawberries, carrots, some root vegetables. They want to be a one-shop stop, but they haven't quite got there yet. But they're moving in that direction.
It's really impressive to see these big players in on this now, right?
Exactly. That's exactly right.
I mean, you espoused this years ago. You should be very happy about that, right?
I'm extremely happy, of course. Like the guys that wrote the RNA article that you just reviewed. They must be very happy to see all these vaccines.
Yes, but you were famous from day one, Dixon.
No, that's not fair. That is not fair.
You have the book. You have the book on vertical farming.
Yeah, well, that's true. I won't deny that.
So you've been an evangelist for a long time.
Yeah, but I try to modify it with a little bit of reality also, because
I understand.
As you'll see my pick of the week, you'll see.
I have to say, when we went to Gotham Greens, I mean, I'd never been in one of those.
It was fun, wasn't
huge. I mean, it's on top of a supermarket, right? And just rows and rows of these greens.
That's right. Basil. They had mostly basil as we were there.
You can buy that stuff around here. I've eaten it. It's really good. And they say they sell everything they make.
That's right.
They grow, they sell. And they have, 21-day rounds of growing, right?
Yeah, it's about, it's more like 6 weeks, but they're now in 3 different cities. So you know that they're doing well. They just quite well. They grow, they harvest, and then they plant, and they do it again all year round. You don't have to worry about the winter. Exactly right.
Although I have to say, we in our garden we put up some of these plastic tents, right?
Yes.
And we harvested parsley and spinach a month ago.
Oh, sure.
And some escarole. It was growing like crazy.
How about that?
I mean, it's not warm. It's warmer than it is outside, right? But it's amazing what will grow even outside under a plastic tent. The problem is harvesting it is hard. It's painful. It's cold. I wanted to do it really quickly because you have to take your gloves off to do it.
But chase out the rabbits first.
The rabbits are somewhere else. They're just... it's too cold for
them. Rabbits are going to learn to fly.
They are.
So that they can get out the vertical
jump higher.
All right, so Chris, we'll do one more round. Chris writes, "This is just my own curiosity. This is not for diagnostic purposes or representative of any trend." And I love that. People tell their stories, and that's really what... Is it possible to have COVID and not test positive on serial at-home rapid antigen tests? As of yet, I have never tested positive for COVID, and I find that extremely unlikely. At the risk of moral opprobrium, I've been going out plenty. We would never judge you, don't worry. New Year's, long weekends, birthdays, buck wild. I'm a 38-year-old vaccinated male with no pre-existing conditions who lives alone and works remotely and skipped Christmas with my elderly family. But I know I've been exposed to Omicron and Delta, but really Omicron is the relevant one here. I'm in Chicago. We're right behind New York in terms of the wave. Literally, all of my friends have had Omicron. Literally, I can name 50 people off the top of my head. And one of them, I was in a broom closet the day before I tested positive. I don't want to know.
Yeah,
Right.
And yet no positive test for me. However, the week leading up to Christmas, I had some vague illness that sure matched the description of Omicron in youngish vaccinated individuals. PCR tests were extremely difficult to come by. See tangential link below. So I had to resort to 3 rapid tests. I had symptom onset Sunday evening, swabbed on Monday, Wednesday, Friday, no positive. Is it possible to have mildly symptomatic COVID but not test positive? I've just not really heard of that happening, and unfortunately I couldn't get PCR as a control. Tangential link, I considered going to one of the many awful, scammy-looking test sites that have popped up. I'm glad I didn't waste my time. And it's an article about that, those scammy test sites. I don't know. Go ahead. Yes, go ahead.
Well, so I have an opinion. I have seen anecdotes, and they don't make data. And I know this is going to go against a little bit of what Daniel said about whether or not you should be doing throat swab for your rapid test. Someone who, their work was insisting that they come back to work, and they kept testing negative by their rapid test until they included a throat swab, and then they tested positive. And that was all before they could get a PCR test or without a PCR test. And so then they were able to say, no, I've tested positive. I can't come back to work. And they had symptoms, and there's some people that are saying that even though the tests weren't licensed for that, the rapid tests that have EUA approval, that if you did a throat swab, then you might get a positive. If you had a lot of tests, you could actually do the experiment, kind of like the way that person did, and do a test without the throat swab, just do the nasal part, and then do the throat swab and nasal. So I think it's possible that, that this person really was positive.
Yeah,
and just tested negative in the rapid tests.
Could be. Also, it could be that he had some other respiratory illness, right? Which it's winter and that happened. There are many other viruses that could do that.
Right.
So, he thinks that he should have been exposed, but who knows? I think it's hard, but I think, it could go either way. Kathy, I think you're next, right?
Anthony writes, a menace to public health. Doctors demand Spotify puts an end to COVID lies on the Joe Rogan Experience. I hope that you remove your podcast from Spotify if they don't act to stop misinformation. So, who was it that just took their music off?
Neil Young, was it?
Yeah, I guess it was Neil Young. Yeah, speaking of Neil Young, took his music off Spotify.
I think he put it back then or something. I don't remember.
Oh, really? I didn't hear the
Neil Young Spotify. Let me look it up.
They were using it at political rallies, which he was upset with because they were right-wing.
Spotify removes Neil Young's music after complaints about Joe Rogan, after he complains of misinformation. The singer decided to leave the streaming service because it gives a platform to Rogan. Huh. So he took his songs off, yeah.
Right.
So this Rogan had Robert Malone on, right? Who just spewed nonsense on the show. That's what the brouhaha is about. And Malone is the guy who says he invented mRNA vaccines, right? You remember that? Have you heard that? Yes.
No.
It's ridiculous. Yeah. Anyway, I think we have to stay on Spotify to counter it, right?
Mm-hmm.
I mean, I know Neil Young protested because he makes the money. So if he takes his songs off, they're probably to say, okay, we'll take Rogan off. But they're not taking Rogan off because he's the world's biggest podcaster. So, and we make no money. So I think we need to counter by keeping our information on, right? That's my thought.
Not taking Rogan off because he's the world's biggest podcaster is criminal, I think.
I don't know why he had... Well, I know why he had the guy on, because he says things that people like. That's what Alan says all the time. He says things people like and they listen, right? And they get millions and millions of listens. I mean, so Lex Fridman is a friend of Joe Rogan's, right? Lex Fridman, I was on his pod. And his pod is pretty popular also. In fact, when I was recording, he got a call from Rogan. He said, I have to interrupt. And he talked to him across the room. I could hear him. Fridman got me on to tell you the right things, and then he gets some wacko anti-vax person on later.
Right.
To present a different... Why? If I told you the story, why do you have to get some... Because it makes money. That's the only reason I can think of. Doesn't make any other sense, right? And he said, I heard him say to Rogan, you should have Vincent on. You should have Racaniello on. He couldn't even pronounce my name.
Racaniello. Nice.
Rich.
Susan writes, hello, TWivers. I'm in Minnesota, so I have you all beat in terms of cold weather. I have been listening to your podcast for the past year and appreciate all your work to help educate the public on these issues. I advise employers on COVID-related workplace policies and vaccine mandates, so I have to dig into the science in order to provide informed advice. Not only is your podcast a fantastic resource, but it has helped guide me in terms of who to listen to and not on science/med Twitter. One expert I follow, aside from all of you, is Professor Akiko Iwasaki, who has been working on mucosal vaccines. I'm hoping you will cover her new study on the intranasal spike vaccine booster, even though it is a preprint. I would like to understand it better and hear your opinions on the premise of this type of vaccine, which would seem to be a game changer. I agree this is an overused phrase, but it might apply here. It gives a link to the article. The article is titled Unadjuvanted Intranasal Spike Vaccine Booster Elicits Robust Protective Mucosal Immunity Against Sarbecoviruses. And it's from Akiko Iwasaki at Yale. And I'm just looking here to see whether this is like in an animal model or what. And I don't see
mice.
it.
Yeah, it's mice.
Mice, okay. While I have your attention, we ought to check this out. I haven't looked at it. While I have your attention, there is also one question I can't seem to get my head around. That is, how to square the notion that if we vaccinate enough people, the virus will eventually not have anywhere to go and die out, with the notion that more vaccination puts a selective pressure on the virus, causing it to evade immunity. This is a line from Apurva Mandavili's article today in the New York Times. Quote, as more and more of the world is vaccinated, evolution will favor forms of the coronavirus that can sidestep antibodies and other immune defenses, end quote. I wonder who the heck this person is.
Apurva Mandavili?
Yeah.
I think trained as a scientist and has been writing for the New York Times.
Okay. I would love to understand. Notice that my first reaction to that is, who is this? I would love to understand this better and as well hear how to stop this from happening. Thanks for all you do. Regards, Susan from Minneapolis. How to square the notion that if we vaccinate enough people, the virus will eventually not have anywhere to go and die out. First of all, that's probably not true, okay? Because I think there is enough
animal reservoirs.
Waning of immunity. There is enough variation in the virus. There is, still infection going on in the absence of disease, so that the virus is gonna be here forever. Okay, like the other human cold coronaviruses.
But you have other
the
animals
difference
too,
between
right,
the
Rich?
excuse me.
You have other animals.
Other animals potentially spill back.
Of course.
Okay, that's a possibility. Possibility of other animal reservoirs. So, so it's not going away.
No.
I would say that once everybody's vaccinated and et cetera, and we have a high level of seropositivity, there's not going to be as much virus around,
right?
I would think. Okay. So, as we've talked about before, evolution of a virus is going to have some proportionality to how much replication is going on. So if there's less virus, there's less replication. That's going to slow things down some, I would think. Okay. I'm just riffing on this. You guys can trash me.
No,
when you're ready. Okay.
Not going to trash.
The notion that vaccination puts more selective pressure on the virus, causing it to evade immunity. Sure. You know, the virus is gonna evolve to evade immunity. The important thing is that there are enough conserved regions in the virus that aren't subject to as much variation, and there's more than just humoral immunity going on, such that even if there's evasion in immunity, it doesn't mean that we're going to have some huge surge necessarily. It just means that the virus is going to keep on cooking. All right. This brings to mind Jesse Bloom's paper that looked at the variation in the human coronavirus, because we always thought that these things were stable. But given what's going on with SARS-CoV-2, he had a retrospective look at what's happened to one of the human coronaviruses and discovered, yes, by God, that thing has been varying over the decades as well. Okay, so it's a balance of these two. I think the high seropositivity is gonna help keep things under control. The immune system is going to help keep things under control, but the virus is going to squirm around and come up with new ideas. And but I'm confident that the high seropositivity and the sort of robustness of the immune response, including a T cell response, will keep us safe.
I think that this statement is an oversimplification because, of course, it's any kind of immunity will be a selective pressure, not just vaccinating. And the idea of sidestepping ignores the fact, as you alluded to, that the variants have been selected for by immune pressures, and we still have, with an ancestral vaccine, we have protection against severe disease, right? So sidestep is not really telling you the whole story, but I think you're right that the common cold coronas, what do? They vary, they infect everyone, they cause mild disease. So in the end, it's not a problem if this virus ends up in the same place. So that's my take.
Yeah, so I think this person has kind of restated what was written a little more strongly than what was written. Because the person says that virus will eventually not have anywhere to go and die out. And I think that rubs us the wrong way, because we know that, for instance, there's places where there's enough people vaccinated against measles, and yet the virus still has somewhere to go and doesn't die out. But, what she's saying is just that as more and more is vaccinated, this is what will happen. But she's not saying as more and more of the world is vaccinated, the virus will go away and die out. So I did finally find where I found something about her education. She has a Master of Arts degree in journalism from NYU and a Master of Science in biochemistry from University of Wisconsin-Madison.
Okay.
And has won some awards in science journalism and has been active in enhancing diversity in science journalism.
Good.
I just think
now you'll see her byline a lot now that you've been.
Yeah. I just think the quote is on its own, of course, but it's an oversimplification. What's happening?
If there's anything wrong with the quote, it's evolution will favor forms of the coronavirus that can sidestep antibodies and other immune defenses.
Yeah, what's the
antibodies? Maybe on other immune defenses, not so much.
Certainly not T cells, right?
Well, It all remains to be seen, but so far it looks like the T-cell response is pretty good.
All right, one more, Dixon.
Patrick writes, Dear TWiV, greetings from Arlington, Massachusetts. I don't understand how there is so little discussion on the relevance of the length of incubation periods to the effectiveness of vaccines. Months after getting any vaccine, the antibodies active in the blood are largely irrelevant because of how much they have been reduced. It's the more rapid activation of the adaptive immune system from prior immune memory that fights off the disease. That takes roughly 5 days on average to ramp up to full gear. The really great vaccines like smallpox, MMR, and polio are against viruses with relatively long incubation periods. Smallpox for 12 days, measles 11 days, mumps 16 days, rubella 14 days, and polio 7 days. The immune memory from the vaccine always wins in those scenarios, even with the inherent variability. But with COVID having only an incubation period, median incubation period of 5 days and possibly 4 for Omicron, and infectivity even before symptoms onset, the immune memory will often lose the race and leave us with infectious and diseased individuals, though protected in time to usually avoid hospitalization and death. Do you see any flaw in that logic? Thank you for your good work, Patrick.
I think it makes perfect sense, right?
Yeah, I never thought about this before. I think this is a very interesting idea.
Yep.
But that's why the respiratory infections are hard, because they have short incubation periods. And
they're infectious before you become aware of them.
Well, that's for SARS-1. It wasn't the case, and it was easy to
I guess that's
curtail that
right.
outbreak.
Yeah, that's right. But this is different.
So this brings up a question that I had, and I think it was when Florian Kramer was on, and he was talking about something about this, about how fast the secondary immunity ramps up, the memory immunity. And I think he was talking about IgG, and I wanted to know, for mucosal immunity, is it also the same lag, or do we know? Because that might make a case for, making mucosal vaccines, for example, if they could react faster.
Yeah, I mean that's the mucosal vaccine boost that was in that preprint. Yeah, But the other thing that these viruses have in common, smallpox, MMR, polio, not only do they have a long incubation period, they have a viremic phase. So polio, all of them infect a mucosal surface and then go somewhere else through the blood. And there they can get nailed by antibodies, right?
Right. So viremic, for people who might not know, means in the blood, a phase of the virus that's in the bloodstream.
So in fact, polio, the inactivated polio vaccine, does not protect your intestines from being infected by polio. You get infected, you shed virus, it gets into the blood, and there, boom, the antibodies nail the virus, so you don't get paralysis. So I think I don't know if it's just the incubation period. I think it may have something to do with viremia. And John Udell mentioned that last time, actually.
Right. And it could be a combination.
Absolutely.
They're not mutually exclusive.
Absolutely. Probably a combination of things. Yep. Okay. It is time for some picks of the week. Dixon, what do you have for us?
Well, my pick stays the same, but my explanation has changed. I originally picked this picture up off of a site which I actually forgot, and I took the picture and I put it on my screen and I left it there because I knew I was going to use it as a pick, but I had thought I took it off of a website for the ISIS, the International Space Station, and Vincent took a look and said, but those guys are standing. How can you be standing on the International Space Station? You're weightless. He was absolutely correct. So Rich looked up this picture and found it on the NASA website. Of course, that's where I originally saw it, and I forgot. But I will just say that because of the dual interest in raising food indoors and escaping to another planet before this one collapses, NASA has reinvested a lot of research in indoor farming. And they were one of the originals to start. And now you see what they've expanded to. And I envisioned something like this being connected to the International Space Station. I think this is a module that could almost go into space right now, screw on just like you would, like jars onto a mothership, so to speak. And you could have an extension of the ISIS with all of this growing that you could feed. How many astronauts are actually at one time on the ISIS, Rich?
Like 6 or 7.
6 or 7.
Usually.
So NASA, originally, when they were looking at indoor growing, they figured that each person needed about 10 square meters of space to raise enough food to keep them alive continuously. So you could just do the calculation and find out how big a container you would need in order to fully satisfy the nutritional requirements of a full load of ISIS passengers as they sped around the Earth and looked at all those wonderful sights. It's quite uplifting to see how far they've come along in their thinking process. And of course, they've had a lot of help. I have a good friend who works out at the University of Arizona. His name is James Giacomelli, and he's done a lot of consulting for NASA. So I imagine what you're looking at is a lot of his ideas included into this design that these NASA technicians or perhaps NASA engineers are now tweaking in order to get nutritionally balanced diets, freshly prepared, and part of the cycle of life on board. Imagine how much oxygen these plants are giving off. That's a wonderful thing, because now your scrubbers don't have to work as hard. And if you really plan this well, you could probably design an ideal unit so that as much CO2 is given off as being is absorbed into the plants, and you could have a perfectly balanced internal ecosystem on your way to Mars. I'm excited
they try
about
that
this.
In Arizona?
Not in Arizona. No, not in Arizona. That's right.
that didn't work. But I have 2 comments on this.
Sure.
The pictures, scrolling down pictures, they show Lane Patterson, who gained valuable experience in indoor farming when he worked at the South Pole Station.
Oh, yeah.
Operating an indoor growth chamber that was partly funded by NASA. And then if you scroll to the very last picture, it should make anybody who read or saw The Martian happy because they show specialized spuds. They show actually growing potatoes.
That's right.
Yeah, thank you, cool. Kathy. I was thinking about The Martian when I saw that.
So the operation of that Antarctica indoor growing system is a project of Gene Giacomelli's, and Lane is one of his graduate students.
Mm-hmm.
So they're all, it's a kind of a tight-knit group, and there's not a lot of people working in it, but the ones that are there are doing wonderful work, and I think that it's got a bright future.
How about kale?
What you're looking at here, you might even be looking at wheat. NASA invested a lot of energy in looking at what it would take to grow wheat, because it was one of the staple crops that you could grow and then store as a grain and use as a supplement to various other diets.
Well, I have said frequently in the past that I wouldn't mind going to Mars, but if my diet is kale, I'm not so sure.
Yep. Yeah,
looks like there'd be a bunch of basil too, though.
That's right.
That's
right. That's right.
Basil and potatoes.
Well, if they made pasta out of the wheat, then you could actually have some very nice combinations. I think.
I'm there. I have pasta. I'm there.
Yep, me too.
Cool. Kathy, what do you have for us?
I picked an article thanks to Justin, who sent me this. It's from Chem and Engineering News, How Pfizer Scientists Transformed an Old Drug Lead into a COVID-19 Antiviral. Oh. And it's how the medicinal chemistry that led to the pill Paxlovid. And basically how it all happened within 2 years. So you talk about fast vaccines. This came along pretty quickly too, but it kind of goes, it seemed like it fit well with today's paper on the biochemistry with RNAs, but it's very medicinal chemistry like, but even I could appreciate it. They, it got to this point and they needed it to be soluble. And so they had to do this. And it's just really fascinating. It's an easy read for the layperson, I think, and worth checking out. It's cool.
Rich, what do you have for us?
So a couple of episodes ago, I think it was Alan who mentioned the Fermi Paradox that I'd never heard of before. Okay. So I looked it up to figure out what this was. Alan alluded to it, which is if there are if there's such a high probability in the universe that there is other life, how come we haven't been contacted? Okay. Or experienced this so far. So I looked it up, and I'm linking a Wikipedia article on this that is really quite good. And I just want to highlight a couple of things from this. So this is the basic thing is, if there is extraterrestrial life in sufficient abundance, how come we haven't been contacted? And this evolves from an event where Enrico Fermi, who was a physicist involved in the Manhattan Project, was involved in a casual conversation in the summer of 1950 with fellow physicists Edward Teller, Herbert York, and Emil Konopinski. Walking to lunch, the men discussed recent UFO reports and the possibility of faster-than-light travel. The conversation moved on to other topics, okay? But then they sat down for lunch, and Fermi says, "Where is everybody?" They immediately understood that based on their early conversation, he was saying, "Where are all the aliens?" Okay? And this goes on from there, and apparently this has been, a conversation that has been going on in many different circles for a long period of time. This article is great because it has a long list of hypothetical explanations for the paradox. One category is that, in fact, intelligent life is rarer than we have predicted. Another is evolutionary explanations, and this is the one that Alan likes, which is that it is the nature of intelligent life to destroy itself, or it's the nature of intelligent life to destroy others. I like this, alien life may be too alien. Okay, we may not recognize it as alien life. Sociological explanations, for example, colonization is not the cosmic norm. Economic explanations, lack of resources to physically spread through the galaxy, for example. Discovery of extraterrestrial life is too difficult, and that I like this, humans have not listened properly. That strikes me as highly probable. Willingness to communicate. Everyone's listening, but nobody's transmitting. Communication is dangerous. Okay, and on and on. So I thought this was pretty good.
Maybe Dixon's an alien.
Maybe he is.
I hadn't thought of that. That's when
I meet him, I'll ask
living
him.
amongst us. Maybe they're. This is fascinating. I just think this is so interesting.
I'm not sure
it's
I would
kind
call
of
this
like
a paradox, though. I object to that word paradox in the sense that you haven't defined the problem well enough to call it a paradox yet. Really. Yeah. I mean, we've only been looking in outer space ever since Galileo's telescope, right? I mean, that's pretty recent.
Yeah, well,
and
we haven't looked very far, though, right?
Wait till you see what the Webb telescope is going to show. I mean, that's going to be a mind blower.
Yeah.
And you still won't see whether there's life on other planets or not, but you can tell whether it's a modified atmosphere. Remember James Lovelock? He concluded there was no life on Mars because the atmosphere kept varying in its composition.
Yeah.
Whereas Earth's atmosphere doesn't vary in its composition, and it's been controlled by life. So 20% oxygen, 76% nitrogen, and CO2 levels, those are all controlled by organisms. That's not a natural geological process. And he argued that Mars's atmosphere changes every time there's a dust storm. And the CO2 goes into the air every time the polar caps melt because it's mostly carbon dioxide. So, and then they kicked him out of NASA, of course, because they said, well, why are you still here? He says, well, that's a good question, actually. So he went on to invent the Gaia hypothesis with Lynn Margulis, which suggested very strongly that life controls the conditions for life on a planet. So I think that Webb has a great chance of making a distinction between all of those Kepler objects that have been found. And now they can look at the atmosphere itself of a planet using Webb, and they can see whether or not it's stable or not, and whether it's containing life-like molecules like oxygen and CO2. So I think those are prescient things. That's going to happen over the next 5 or 6 years, I think. And then we probably won't call it the Fermi paradox any longer. We'll probably call it the affirmation of life no matter what.
That comes then under the heading of humans have not listened properly.
Or been able to listen properly, but we do now. And isn't it amazing? Don't you think this is fascinating? We have spent how many billions of dollars, Rich, to make the Webb telescope?
Lots.
Billions. The Hadron Collider, billions. What practical knowledge would you ever expect to gain from either one of those instruments? And the answer is, we don't know. And
but we funded it anyway. Good for us.
You know what? That's why Vincent's comments about basic research are so important, because we're still intelligent enough to want to know, period. We just want to know.
Yeah. We have the right intentions, although
that's
not often the money.
That's true. But eventually, if it's a good enough question, we find the money.
I mean, the telescope is out there on its own. There are no people on it. So
that's correct.
I think if there were something somewhere else, that's what they would do, because they don't want to send. It takes so long to get somewhere, right?
It took a month to go 1,000,000 miles.
Humans aren't old enough to have gone anywhere. And
but the radio signals, Vincent, the radio signals have been going out ever since there was radio. And that's, into other galaxies now.
I don't know. Look, I don't know. I just think it's too far away. It's not happening.
You're probably
right. I have to say, Kathy, after several previous tries, the astronomy picture of the day is now permanently installed as my Chrome homepage, and I have to say that it's really cool to be reminded every day of just how vast this all is.
Is that the Hubble Space Telescope Deep Space?
It's all kinds of stuff. It's today it's a picture of the surface of the moon.
That's right. The one that really strikes me is the ultra deep field view of nothing, and then they saw everything.
Yeah.
In a little postage stamp square space. They saw something, and how can there be anything there? Of course there's something there, you dope.
And the Webb telescope made it, right? It's orbiting its Lagrange point.
That's right.
And doing the fine adjustments on each of its mirrors.
It's all going to work.
Unbelievable.
All going to work.
Cool stuff. Yeah, Rich, you have a Tinkerbell behind you now.
I've been looking at this, wondering when Tinkerbell was going to show up, and there she is.
So that's your thing, Kathy. You called it a Tinkerbell on the wall. Yeah. My pick is a book by Joe Graves and Alan Goodman called Racism, Not Race, Answers to Frequently Asked Questions. And earlier this week on TWiV, we had Joe Graves join us, and he told us right away he is the first African-American in the U.S., or anywhere probably, to get a PhD in evolutionary biology.
Really?
Yes. And you can argue with him, because I'm not.
No, I'm just very surprised. That's all.
Yeah. Well, that's part of the problem, right? It's not equal. Anyway, I got a PDF of this book, and it's, there is no race. There is racism. That's the whole point. And this has been known for many years. But anyway, the book is set up as frequently asked questions. So, for example, the introduction, what is race? Race is real and not real, huh? How many races are there? What do geneticists mean by the structure of human variation? What is racism? Do the ancient Egyptians, Greeks, and Romans have a concept of race? What changed in Spain in 1492? So really good. What's the differences between bigotry, bias, prejudice, xenophobia, ethnocentrism, and racism? Is anti-Semitism a form of racism? My doctor's office form asks for my age, sex, and race. Why does it ask for my race? It's wrong, right? Because there are no races. So I really think it's wonderful. I think everyone needs to read this, but they won't, because as he will say, our world is based on Europeans, essentially, which are mostly white, and that's what runs everything. And we have to fix it, but it's not going to happen quickly. And part of it is just defining all these issues. So I really like it. I think it's a great book, and
cool.
I suggest everyone read it. It's published by Columbia University Press, which is very nice. I think I'm proud that they published this.
Do they either author have connection with Columbia?
No, they don't.
Good.
But the press picked it up, which is really good. So he was great also on TWiV. So it hasn't come out yet. It will come out sometime next week, but really good.
I've got this little thing that Alan introduced me to, that when we go to Amazon or have a book up, it automatically gives me an opportunity to go to my library and order this book. So it is now on hold.
Now, one thing that has changed now, I set up an Amazon business account for 501(c)(3). So you can't do affiliate links there anymore, which is too bad because you can only do them on a personal account. So this link that I'm going to give you is just a link. But anyway, it's a cool book. And we have a listener pick from Jay, who writes, since you got a kick out of furniture restorations, I thought you might like this channel of art restorations. Jay is in Melbourne, Australia. So it's called Baumgartner Restoration on YouTube. Yeah, and they tell you how to restore various things. It's very cool. Old master painting conservation, restoring a Baroque painting, wood. Very cool. Thank you, Jay. I'm sure I'll love it.
If you had to guess which kinds of paintings were easy to restore and which ones weren't, would you pick the modern painters or would you pick the masters?
Oh, I have no idea.
Well, it turns out that
from the way you asked the question,
yes.
It's going to be the modern guys,
no,
right?
No. It's just the opposite. The masters all used reliable pigments in oil. They're easy to restore. They're absolutely stunning when they're restored and that sort of thing. I had a friend who actually did that for a living. And then you get these modern painters like Mark Rothko, who used combinations of car lacquers, house paint, and squeezing tubes of other kinds of paint in it and mixing it all up and then painting with it. Try to restore that sometime. And you don't even know what it's made out of. So
I know. But it's art, Dixon, right?
It's the old ones that are the easy ones.
That's interesting.
Yeah.
Now, the restoration shtick is interesting. I recently saw a video about a guy who got a pair of work boots from like 1900 and restored them. They were a mess. Really? And this guy's a shoemaker. He had a shop, and he went through what he did.
I'll be darned.
And at the end, they look great. But why would you do that? I guess just restoring them. I mean, he basically has a new sole, a new heel, and the leather is all fixed up, and he did something inside it as well. But just seeing how you do this is quite interesting.
I would agree.
Anyway.
I would agree.
But, the before and after are stunning, right?
Yes.
But you probably don't wear them because you're afraid to mess them up. But it's pretty cool. All right, that is TWiV 860. Show notes are at microbe.tv/TWiV. You can send us questions, comments, pics, TWiV@microbe.tv. Please send them there, not to the other various emails we have because it's too hard to keep track of them. TWiV@microbe.tv. And if you want to send an email to Daniel, that's Daniel@microbe.tv. Don't cross send because it's too confusing for me. I have very simple means and it's too hard to sort out. If you like what we do, consider supporting us. Microbe.tv/contribute. And we are a 501c3, so your contributions are tax deductible. We really appreciate your support. Dixon Despommiers at Racaniello.org, thelivingriver.org. Thank you, Dixon.
You're very welcome. It's a pleasure to, once again, participate in such an interesting discussion.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He is currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Get vaccinated.
And I am Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.